fellow classmates, Miss Lee and fellow classmates. As you know, today we are doing the presentation on photolysis. I'm Hari and I will be your narrator today. As you know, you do not need further introduction as you know all these people. Okay, well, today we'll be exploring photolysis. What is photolysis? From the word photo, you could infer that it is something to do with photosynthesis. Well, that's right. Photolysis or photodecomposition is a chemical reaction in which a chemical compound is broken down by photons. Photolysis is part of a light-dependent reaction of photosynthesis. The general reaction of photosynthetic photolysis can be given as H2A plus 2 photons, which is light, is equal to 2 electrons, 2E plus 2 hydrogen and A. Where light is used to split, photon is the light which is used to split H2A into this compound. Photon is as you know, based on the knowledge that you have learned from chemistry, you know that when photons travel, when electrons travel, they release. Let's make this as electron. Okay, when electron travels from one shell to another, from the top to bottom, they emit certain energy known as photons. Okay, so it's actually the energy released when electrons travel. The effectiveness of photons of different wavelength depends on the absorption spectra of the photosynthetic pigments in the organism. Chlorophyll, which is a pigment, absorbs light in the violet blue. This is violet blue. Okay, violet blue photons. Yeah, violet blue and red parts of the spectrum, while accessory pigments capture other wavelengths. Accessory pigments are other pigments such as carotenoids. Okay, now Siche and Lucien will be explaining to you more about photopigments.
Now let's talk about photopigments. Photopigments are unstable pigments that undergo a chemical change when they absorb light. Photosynthetic pigment converts light into biochemical energy, and examples are chlorophyll, carotenoids, and phycobilins. These pigments enter a high energy state upon absorbing a photon, which they can release in the form of chemical energy. These can occur via light-driven pumping of ions across a biological membrane. For example, in the case of the proton pump, bacteriorhodopsin, or via excitation and transfer of electrons released by photolysis. In chloroplasts, the light-driven electron transfer chain in turn drives the pumping of protons across the membrane. Accessory pigments are light-absorbing compounds found in photosynthetic organisms that work in conjunction with chlorophyll A. They include other forms of these pigments, such as chlorophyll B in green algae and higher plant antenna, while other algae may also contain chlorophyll C or D.
Okay, then now it's my turn. In photolysis, there is also photolysis of water, which is also part of photolysis, which is similar to photosynthetic photolysis, where molecules are broken down into smaller units through the absorption of light. But the difference here is that water molecule is broken down using the light, sunlight, into hydrogen and oxygen. You can refer to the notes if you think we are not reliable, but we are. Okay. Yeah. So that's the photolysis of water.
The hydrogen then combines with a hydrogen acceptor called NADP to form a reduced hydrogen acceptor, NADPH2. Nicotinamide adenine dinucleotide phosphate or NADP plus is a cofactor for some dehydrogen gases. In addition, chlorophyll makes energy available for regeneration of ATP from ADP and inorganic phosphate. This process is called photophosphorylation. Adenosine 5-triphosphate is multifunctional nucleotide and is most important in cell biology. As a coenzyme, that is the molecular unit of currency of intracellular energy transfer. In this role, ATP transports chemical energy within cell for metabolism. It is produced as an energy source during the processes of photosynthesis and cellular respiration, and consumed by many enzymes and a multitude of cellular processes including biosynthetic reactions, motility, and cell division. Adenosine diphosphate, abbreviated ADP, is a nucleotide. It is ester of pyrophosphoric acid with the nucleoside adenosine. ADP consists of pyrophosphate group, the pentose sugar ribose, and the nucleobase adenine, and is an important energy transfer molecule in cell. ADP is a product of ATP dephosphorylation by ATP gases.
Okay.
The hydrogen held by NADPH2 and the energy held by ATP at the end of the light stage are essential for the use of carbon fixation, the second stage of photosynthesis, which will be covered later. Thank you for your kind attention. We hope you have a good time.
And we hope you learned something. And if you think we are not reliable,